basic mechanisms of this adjunct Ayurvedic therapy. So this has come up with CCRAS, the Central Council of Ayurvedic Research and Biotech Research Innovation Council, NII, and also the ministry institutes under Health Ministry like JIPMA. Dr. Sonali is already here, and also under I serve Pune and CCRAS. Next slide, please. Yeah. So cachexia, we won't, in the interest of time, I'll skip. So essentially, this is the increased energy expenditure, reduced muscle mass, or decreased energy output. These are associated with immune dysfunction, metabolic dysfunction, malnourishment. And next slide, please. So when the cachexia and TB is a vicious cycle. So in TB, you have a cytokine activation issues, abnormal protein metabolism. In cachexia has other things, and when both are together, this is a vicious cycle. And there are very few studies which have addressed this issue. But this recent study which has come up is the failure to gain weight, 5% body weight within 2 months is strongly associated with the modality. So that is why to understand the cachexia can help us in better designing better TB cure. Next slide, please. And this is, as was told by Dr. Acharya. So this is a study with a very significant number, 250 new tuberculosis patients will be put on the Ayurvedic regimen along with standard anti-tubercular therapy, and there will be a control arm which is no Ayurvedic treatment, and there is a smaller group just to have molecular studies with the TB patients with the drug, and this is the standard encompassing the parengenamide and ethambutol, and 4 months and 2 months, and there are healthy controls. Next slide, please. And these are the objectives, like how does the body composition changes in cachexia, how the interorgan crust-up, which is the important part, the cachexia interorgan crust-up plays a very important role, and what are the molecular mechanisms of the cachexia, and how affect immune response, which is the more important component, that's why Greek NII is involved, and how energy metabolism, how is Ayurvedic intervention in the molecular details, we can find out how Ayurvedic intervention is helping, and the generation of models for TB cachexia for testing the effect of Ayurvedic intervention. Next slide, please. So this is some of the studies, DEXA scan and MRI, which are the details for the JIPMA, and this is being done at JIPMA, and all the these things. In the interest of time, I'm not going into the detail. So next slide. So here there are some advanced molecular techniques, which is the mass spectrometry based protein characterization. This is MRI and SRI assay to determine these organokines which are determining the interorgan crust-up during cachexia. They are being studied, and of course the metabology like metabolomics will also be used, and single cell like transcriptomics, which can characterize immune cells, which is an important component of this study. So with this, we will be able to get which are the cell types involved, how immune cell is playing a role, and which are the molecular players in this process. Next slide, please. So this is analyzing immune dysfunction. You will also have immune phenotyping using flow cytometry, and specific TB specific T cells will be characterized by ex vivo activation of T cells, and metabolic effects of the immune compartment will be seen. It will be metabolic chamber with this study will be done. Next slide, please. Yeah, so these are the expected outcomes. Of course, establishing the efficacy of the, it is already known, but in terms of the Ayurvedic adjunct therapy, how it is helping. In addition to that, if we can characterize several organokines, they could be used for future drug targets, and antibodies for these organokines, it can be developed, they can be used for therapy. So these are the expected outcomes of the project. Next slide, please. So with this, I would stop. Thank you.